Hello, everyone. Welcome to now our fifth lecture in the series. Um, Today's topic is going to be differential expression analysis. So this kind of builds off of our past two lectures on normalization and hypothesis testing. So just to start off with a quick overview of today's content, we'll begin with just a brief introduction of, what is differential gene expression for those who aren't familiar. So we'll talk about what it's used for and how it fits into the RNA-seq workflow. And then most of our time today will be spent going over these three algorithms for differential gene expression. And then we'll touch on some helpful data visualizations for visualizing these DGE results. And then we'll end with just a brief discussion of DGE methods for single cell RNA-seq data, which is kind of a newer area. Okay, so what is differential gene expression analysis? So usually in biological studies, we have different biological conditions that we're interested in and we want to compare them. So for instance, there may be some cells treated with drug A and other control cells without any treatment. And ultimately, if we want to understand the biomolecular differences between these conditions, then it can be very informative to look at the gene expression profiles of these two conditions. So what researchers will do is they'll take a few samples from each condition and quantify the transcription level or how abundantly a gene is being expressed. And usually this is done with microarray or RNA-seq experiments. And then for each gene, they'll test whether the abundances are equal between the two conditions using some statistical tests. And we call these significantly differentially expressed genes or DEGs. So those are the genes we're interested in. They're likely to be relevant to the condition that we were originally studying. So that's kind of the process of differential gene expression analysis. So to apply this DGE analysis to RNA-seq count data, before we can actually do that statistical analysis, we need these RNA-seq processing steps that hopefully these terms look familiar to you. They were covered in the earlier alignment and normalization lectures. So these steps will essentially transform those raw sequence reads, will be mapped into those gene or transcript counts. And then we need this important normalization step to be applied to the counts. So different DGE algorithms, they take in different inputs depending on what's already built into the package that you're using. So for some of these methods, you might not need to do your own normalization. So DESeq2 and Lima are both methods like that. They have their own normalization that gets implemented when you apply the function. So you're going to want to feed in unnormalized read counts to these two methods. But there are other packages out there that are popular that might expect a specific kind of normalization, such as like RPKM or TPM or something. So that's just something to be aware of, depending on the method you're using. You want to expect different normalization methods to be appropriate. And then I also just want to note that this workflow that's sort of loosely outlined here is what an RNA-seq differential expression workflow will look like. But differential expression analysis in general is a method that you can also apply to other data types. So it's something you can do for protein expression and other omics data. But those data types behave very differently, so they'll have their own specific pre-processing and analysis tools. So yeah, for today's lecture, we're just going to assume that all the data we're dealing with have already gone through that alignment and quantification pre-processing steps. So our starting point is going to be the gene count matrix, which should look something like this. So we saw this in last week's lecture, where we have the rows representing the genes, and then the columns are the counts for each sample. And if we want to compare the counts between samples, the naive assumption might be that we can just look at the numbers. So say if sample 2 has higher counts than sample 1, then we might sort of naively assume that this gene was expressed more highly in sample two. But due to technical variation across samples and differences in library size and composition, in order to actually make that comparison, we're going to need to normalize these samples. So this is just going to be a brief review of normalization. So yeah, these count normalization methods, they're typically aiming to account for at least one of these three things. So there's sequencing depth, also called library size. So this is the number of reads per gene. Then there's the length of the gene, so how many base pairs each gene is, and also RNA composition across the samples. And as I mentioned before, different differential gene expression methods implement different types of normalization. So for DESeq2, this method uses the median of ratios normalization, which takes into account the differences in library size. And then Lima, on the other hand, this method was actually originally developed for microarray data. Lima comes with this VUM analysis method, which transforms the RNA-seq counts to fit Lima's distribution assumptions for microarray. And then some other common normalizations you might see are the reads per kilobase per million reads mapped, so RPKM. So this has both library size and gene length normalization. And then TPM is another one. It's very similar to RPKM, just a difference in the order of operations. But yeah, some other algorithms might use RPKM or TPM, so you might see these out there. But for today, for both DESeq2 and Lima VUM, these two methods have their own library size normalization that's implemented. So the data that we input to these methods should not be in any of these other normalized forms. Okay, so as you're probably already becoming aware of, there are a lot of different statistical algorithms that can be used for differential gene expression. So a big question is, how do we really choose which one is best to use? And the answer is that it really depends, and it depends on a number of factors. So not only the nature of the data that you're working with, but also what the goals of your analysis are. So like the signal size that you're expecting to detect, and maybe your desired control over the false discovery rate. So one of the main differences, at least between each of these DGE algorithms, is that they each make slightly different assumptions about the underlying data. So for example, a log t-test is going to assume that the log counts follow a normal distribution, whereas DESeq2, like I mentioned, I think this was covered last week, it assumes the counts follow a negative binomial distribution. And then a method like Poisson-seq assumes a Poisson distribution, etc. So you're going to want to be aware of what distribution best characterizes your data, and then choose a method that appropriately captures that behavior. So for example, if you had collected data from technical replicates, so these are repeated measures from the same samples, you might choose a method that assumes the Poisson distribution for the read counts. But if you have biological replicates instead, which is usually what's more common, so these are measurements taken from different samples under the same biological conditions. So then in that case, the variability between your samples is going to be much higher. And you might prefer to use the negative binomial distribution to capture that element of overdispersion. And that kind of brings us to another important choice that you'll have to make for this analysis, which is how many replicates do you want to include in the experiment? So to offer some insight on that, this figure on the right here comes from a study by Church et al. That compares different DGE methods in detecting different signal strengths across these 3, 6, 12, and 20 numbers of biological replicates. So what we see here is on the left, these bars are indicating the false positive rate, and then on the right is the true positive rate. Um, and then, so this first column here is the detection of log fold change greater than 0, and then greater than 0.5 and greater than 2. So generally, 3 replicates here is considered to be the bare minimum for RNA-seq studies. And you can kind of see if you want to pick up on these genes that may not be very strongly differentially expressed, so they have small log fold changes. Most of these methods at this limited sample size can only detect these positives at 50%, so the 50% true positive rate at most for these methods. And yeah, so that essentially just means that less than half of the truly significant genes are being marked as differentially expressed under these analysis conditions. So for most researchers, that might not be quite good enough. And generally, the reason we care so much about the number of replicates is because all of these statistical algorithms, they're trying to estimate the mean invariance of some parameter, like the log fold change from the data. And it's very challenging to do that when we have these small replicates, and that leads us to these low true positive rates or higher false positive rates. So going back to this figure, if you kind of just increase the sample size from 3 to 12, we can kind of see how this really greatly improves the ability to detect those truly differentially expressed genes. So this is sort of the effect that increasing your sample size has in this situation where you're looking for genes with potentially smaller log fold changes. But if we look over on this right column here, oops, for these, if we're trying to pick up on a bigger effect size, so a log fold change greater than 2, then here we can see the true positive rate is not as sensitive to these changes in sample size. So essentially, if you're trying to detect genes with lower fold changes, you should be a bit more concerned about having enough replicates compared to if your goal was instead to detect only these higher fold change genes. So yeah, so in the end, it's really just going to depend on your data and what your aims are. So for today, the 3 differential gene expression methods will focus on our DE-seq2, LEMA, and then the characteristic direction method developed by the Mayan lab. So these are all 3 different approaches. So we'll just discuss sort of the broad details of each of them one by one.